photosynthesis part. Did we completed this one or not? This is screen which is visible to your screen. This slide. Photosystem, did we complete it or not? It was there in last class. No one is able to tell me. Okay, no issue. Let's start from here again. There is nothing wrong to revise this PPT. Okay. So photosynthesis factual thing I have told you that I know that let's restart from here. So chloroplast where photosynthesis takes place, right? So this is the chloroplast where actually photosynthesis takes place. Means this is the site of photosynthesis. So exactly where the photosynthesis takes place in the chloroplast, site chloroplast. At what part? At what particular point? Specific point, right? Okay. Look. So as we know that the photosynthesis takes place in the green plant, right? So in the green part of the plant. So where in the green plant part of the plant? Basically, the leaves are meant for the photosynthesis. Inside the leaves, so there are particular kind of the leaves. They are the modification of the parenchyme, right? So these leaves are specifically responsible for the photosynthesis. So their job is to perform the photosynthesis. These leaves are known as the mesophyll leaves. They are the mesophyll leaves. So mesophyll leaves are the responsible for the photosynthesis. They are designed for the photosynthesis. Inside the mesophyll leaves, that is very important. This is a question it has been asked. You will get this kind of the question in your practice the practice question paper mock test which I am giving you, because I am the person who is designing those mock tests, right? So look, in the mesophyll cells you will find the chloroplast at the peripheral region, means outer region, right? The protoplasm, and there will be, so why are they at the outer surface, outer region? So that they could get the better light and perform better photosynthesis. So these mesophyll cell will have the chloroplast, right, at the peripheral part. Clear? Mesophyll cells are meant for the photosynthesis. Now, inside the mesophyll cells, there are a number of the chloroplasts. Let's see what is the function of. This is the diagram of chloroplast. So chloroplast is made up of two layers: outer membrane and inner membrane. So outer membrane and inner membrane, between these two membranes, there is a space that is called intermembrane space, right? Inside the chloroplast, it is filled with the gel-like substance, which is called stroma. In the stroma, there is these piles of the coin, right? These structures. These structures are known as what we call them. These structures are known as granum, right? So in the stroma, there are these structures. These are structures are called granum. All the granums remains connected with the other granum, right? With the help of a structure, and what we call this structure is known as stroma lamellae. What we call that? Stroma lamellae. So stroma lamellae is the structure which connect a grana to another. So these grana are made up of small coin-like structure. These coin-like structures are known as thylakoid, right? So they are called thylakoid. The photosynthesis takes place in two parts: the dark reaction and the light reaction, right? Light reaction takes place in the presence of light. Dark reaction takes place in presence or absence of light at the both of the places. So light reaction takes place in grana. I have written over here. Dark reaction takes place in stroma. What is the light reaction? So we divide photosynthesis in two parts: light and dark reaction. What is the light reaction and what is the dark reaction? Let me tell you. The dark reaction is called fixation of CO2, right? And what is the light reaction? Light reaction is also known as the photolysis of water, means breakage of water molecule, breaking of the water molecule. It is also known as the Hill reaction. So light reaction is also known as the Hill reaction. This is nothing but photolysis of the water, right? So these are the thylakoid, right? In these thylakoid, you can see these are the thylakoid. In these thylakoid, At the peripheral part, there are certain chloroplast molecule, right? Sorry, chlorophyll molecule. So these are the chlorophyll molecule. Chlorophyll is a magnesium compound. That question also have been asked. Very simple question, but this question have been asked many times. So chlorophyll is a green color magnesium compound. Now you can see the part of thylakoid which touches with another thylakoid. These are called appress part. Guys, remember these the part of the thylakoid which is in touch with or in contact with the another thylakoid. These are called appress part. So they are called appress. And the part of the thylakoid which are not in touch with another, these are called non-appress part, right? So there are appress part which are not in touch, means outer side, and the appress part. In appress part, the photolysis of water takes place, right? In these parts, right? Photolysis of water takes place. In photolysis of water, two molecule of water breaks down. Due to breakage of two molecule water, 4 hydrogen ions are released, oxygen molecule is released, and 4 electrons are released. This is called photolysis of water, which takes place in appress part, right? And in non-appress part, there is some another cycle goes on. So this is the structure of the uh, functional part of the chloroplast. Is it clear? What takes place? Where takes place? Okay,
In
fine.
CO2 fixation, what happens?
Now we'll tell you that is a Kelvin cycle. CO2 is fixed and it is converted into glucose. Means CO2 is taken in the gas form. It is converted in some another compound, which is a stable compound. What it?
Yes, sir.
Look, what is the difference in the light and the dark reaction, or what is the correlation between them? Now let me tell you. What is the correlation between the light and dark reaction? Here you can see the light reaction, that is a breakage of water molecule. If that takes place in presence of light. So light reaction goes on only in daytime because this reaction means photolysis of water, it requires light. Due to this light reaction, you can see formation of ATP takes place, which is energy currency, and formation of NADPH takes place. That is also, what is that? That is also energy currency, right? So formation of energy takes place, ATP and NADPH. This ATP and this is called dark reaction. You can see this is the called dark reaction. Now ATP and NADPH is utilized in Calvin cycle. Calvin cycle, that is a dark reaction. And what we call a light reaction? I have already told you this is called Hill reaction. Hill reaction. So that takes place. So what happened? This reaction do not require light. So it goes on the day and night both. So dark reaction goes on day and night both, but light reaction goes on only at the daytime. What happened in the dark reaction? Fixation of carbon dioxide takes place. Calvin cycle goes on. Now, due to the Calvin cycle, formation of NADP plus and ADP takes place, means they are utilized. And again, this NADP is converted into NADPH. This ATP is converted to ATP, right? And due to Calvin cycle, what happened? Due to Calvin cycle, formation of sugar or food takes place, right? Means formation of glucose takes place due to this dark reaction. Due to this light reaction, formation of O2 takes place, formation of NADPH takes place, NADPH2 takes place, and formation of ATP. One very important thing which you have to note it down at anywhere in this chapter, one GTP is equal to one ATP, both are the equivalent kind of the source of energy, but one NADPH is equal to 3 ATP. One FADH is equal to this energy equation you need to know. Tell me, is it clear or not?
Yes, sir.
Next, look. This is the actual diagram of thylakoid. So inside thylakoid, at the membrane of thylakoid. So thylakoid is like, you can see it's You must have seen some toffees where the outer cover is hard, inside there is some liquid kind of spilled. So you can see it's like choco pie. The outer cover is hard, not choco pie actually. The biscuits where the chocolate is remain spilled. I forgot the name. So inside the outer cover, this is made up of membrane. Inside there is some fluid. This is called lumen. So in this membrane, there are two kind of the photosystem. PS1 means photosystem 1 and PS2 photosystem 2. PS1 is basically called P700 and PS2 is called P680, right? So this is the membrane. I have told you what is the appress part, what is the non-appress part. Appress part which remains in the touch with another thylakoid. Look, in appress part there will be photosystem second, means P680. When I talk about the photosystem second, means we are talking about the P680, right? In appress part there will be P680, and this appress part means photosystem second. In these part, the oxygen breakage takes place. Oxygen evolving complex OEC is found, which help in breakdown of the water, right? Formation of oxygen takes place. So breakage of water takes place and oxygen is formed over here, right? In non-appress part here, you can see. In non-appress part, what happened here? Some other compound are found. We'll discuss those compound. Photosystem 1 is found in non-appress part, right? You can see photosystem 1. In non-appress part, it is found, right? Non-appress part, there is also enzyme is found that is called ATP synthase. What is the function of ATP synthase? Basically, ATP synthase, formation of ADP takes place, formation of ATP takes place from ADP. Means this ADP combined with one phosphate and make ATP. Means production of energy takes place with the help of ATP synthase enzyme, right? So this is the structure of the thylakoid. Is that clear? Photosystem means one means P7, just found in non-appress. Clear? So evolution of oxygen takes place, this breakage of water takes place in appress part. Now, what are these photosystems that I want to tell you? What are these photosystems? Photosystem, that is very important. The question you will get question from here. Even you can get the question that oxygen evolution takes place, appress part, non-appress part, both of them and all that, right? Look, photosystem is made up of 250 to 400 pigment molecules. Means molecule of chloroplast and chloroplast like molecules, like chloroplast like molecules. So this photosystem, like what is the composition of photosystem? It is made up of 250 to 400 pigment molecule. The center of photosystem will be made up of two things. The center of photosystem is called reaction center. All the energy from the surrounding will come at this point at the reaction, right? Reaction center will be made up of two things. Primary electron acceptor means finally which will accept the electron, and a reaction center which is made up of a special kind of the chlorophyll A molecule, right? So this is made up of chlorophyll A molecule and primary electron acceptor. In both of them combinedly, they are called reaction center, right? Around the reaction center, there are some antenna molecule, pigment molecule. These will take the photon. They will absorb the photon. This photon will be captured by the chlorophyll molecule, right? This chlorophyll molecule. They will be chlorophyll and carotenoid. Finally, they will transfer their energy to reaction center. Reaction center will give energy to electron transfer, primary electron acceptor, right? So this complete system is called PS1. Now, what is the PS1 and what is the difference between this is the photosystem basically. The both PS1 and PS2 look like this one, right? Now, what is the photosystem 1? What is the difference between two? Look, PS1 is found in non-appress part, right? Found in non-appressed part. Then more chlorophyll A molecules are present. Chlorophyll B and chlorocarotenoid are found in less amount. Photocenter is called LHC, light harvesting complex 1, LH1, an electron carrier. Reaction center is made up of chlorophyll A molecule, that is called PC100. Both cyclic and non-cyclic photophosphorylation goes on here. I will tell you what is the cyclic and non-cyclic photophosphorylation. Then PS2. Now the PS2 is located in appress part. We have discussed that. It contains chlorophyll A, B, and carotenoid molecule. Chlorophyll A and chlorophyll B are in equal amount. Both are found in equal amount. Equal amount. Both are present in equal amount. Photocenter, oxygen evolving complex, LHC2, that makes the photocenter, that is the reaction center. Photocenter is made up of, the reaction center is made up of a dimer of chlorophyll A molecule. Dimer means two chlorophyll A molecule. It contains oxygen evolving complex. Now this is very important. Oxygen evolving complex, which is present at the photocenter second, it is made up of magnesium ion, calcium ion, and chloride ion. Oxygen evolving complex that it releases the electron during the photolysis of water, right? So this is the PS1, PS2. Tell me, is it clear or not? Photosystem clear or not? Tell me, guys, is it clear or not?
Yes, sir, clear.
Alan, clear? Hanifa, Farad, is that clear? Mamuna, Aksa?
Yes, sir.
Yes.
Very good. So you have to remember the composition. That is the question. Let me tell you what is the question. What kind of the question you will get? How many molecules are there? Which of the following is correct about the photosystem? That kind of the question. Which of the following true about the photosystem 1? Right? This kind of the question. You have to remember all these points. Electron transport chain. Right? This is the chain which help in providing the energy. Electron transport chain can be of two types, cyclic and non-cyclic. Right? In non-cyclic phosphorylation, this is called ZST. Right? Now, what is the non-cyclic photophosphorylation? So, what is the photophosphorylation? Any one of you, please tell me, what is the photophosphorylation? What is the phosphorylation? Any one of you, I will appreciate your answer. Please try it. I'll be very happy if you'll answer me. What is the phosphorylation? Who is going to tell me? Safullah, please tell me. Guys, tell me. No one is.
So it's attachment of phosphate ion to a molecule.
Good try. Farad,
Yes, sir.
tell me, what is phosphorylation? Mamuna, tell me at least, what is the phosphorylation? Mamuna, did you take in 11th class from me? Are you the same Mamuna who have taken 11th class from me? Was you there in 11th class?
Yeah.
Okay, then the biggest responsibility on your shoulder, because I am sure I must have told you.
It's like an attachment of phosphate group in to a molecule or ion. Something
It's
like that.
You are close to the answer. Look, remember, guys, what is a phosphorylation? in phosphorylation, what happened? Then I tell you what is the photosynthesis. ADP molecule, that is adenosine diphosphate, it will make a bond with Pi, means inorganic phosphate, and the formation of adenosine triphosphate, ATP, will take place, right? This is the energy currency, means formation of energy will take place with the help of enzyme, right? Which enzyme we have discussed just a few minutes before, right? With the help of ATP synthase enzyme. ATP synthase enzyme. This reaction is called phosphorylation, right? So let me elaborate this reaction for you. I want you, what is the adenosine diphosphate? This is the adenosine sugar. Adenosine is a pentose sugar, right? It is adenosine sugar, right? Now what happened? That is ADP, diphosphate. So there are one phosphate. I am making the bond like this way. This, it shows the weak bond, means they can be. Now one phosphate, inorganic phosphate. Pi means this inorganic phosphate. ADP, so this is the adenosine sugar and it have two phosphate. So it becomes ADP, adenosine diphosphate, right? Now this is inorganic phosphate. Inorganic phosphate will be taken from the protoplasm. Now what will happen? This inorganic phosphate will combine. Now there will be 3-phosphate. And we know when the bond formation take place, energy is stored. And when the bond will break, then energy will be what? Produced.
Release.
Right? Energy will be released. So this bond will break down and it will release energy. That's why when we say that ADP become ATP, means energy formation is taking place. When ATP will break down into ADP, means energy production is taking place. Is that clear or not? Tell me. Is that 100% clear?
Yes, sir.
Okay. Now what is photophosphorylation? Phosphorylation means formation of ATP. Photo means light. When formation of ATP takes place in presence of light, this is called photophosphorylation. Clear?
Yes, sir.
Keep it in mind, right? This is very important. These are the small concepts, but they are very important. Now, what is photophosphorylation? So, obviously, in presence of light, non-cyclic photophosphorylation. Why non-cyclic? Because electron will start its journey from one point and it will end at the another point. That is non-cyclic. It is not taking cyclic. In cyclic, the electron will start its journey from the one point. It will come back at this point again. That is cyclic photophosphorylation. But in the both are the phosphorylation. Non-cyclic, electron will come from one point and go will another point. Vanished. Consumed. In cyclic, electron will travel from one point and come back at that point. But both are the phosphorylation. Means the formation of energy or ATP will take place at both. This graph look like jet. That's why we call it jet scheme. Now come to here. Easy. You must have gone through. You must have already studied it. Look here. Photolysis of water taking place. P680. That is the PS2. Right. Why this is PS2? In the reaction sequence, this is the first member. But why it is PS2? Because it had been discovered later. The first member, first photosystem discovered, that was the P700. That's why we call it PS1. Get the logic, right? This is called PS1 because the P700 or PS1 had discovered first. Then after that, PS2, which is discovered, that is called P600. So look, reaction start from P680, right? What will happen here? Oxygen evolving complex is present, right? What will happen? Water molecule will break down. Due to breakage of this water molecule, oxygen will be released. One oxygen molecule and 4 hydrogen molecules, right? Remember, these 4 hydrogen molecules have gone over there. I will tell you where they have. Now, what will happen? The hydrogen molecule plus electron will be evolved, right? Electron will be released. Now, these electron will be taken by the reaction center, right? Due to light, right? Reaction center will give energy An electron will be taken by primary electron acceptor. This primary electron acceptor, the electron get excited due to the energy. Now what will happen? This primary electron acceptor will receive the electron, and electron will reach to the pheophytin. You have to remember only those names which I have encircled. Pheophytin. From pheophytin, this electron will reach to the plastocunone, right? From plastocunone, this electron will reach to the cytochrome B6F, right? Cytochrome B6F, this one. When electron will reach to the cytochrome B6F, formation of ATP will take place from ADP. You can see here. Formation of ATP will take place, means energy will be generated. So from pheophytin, it will reach to the plastocurine. Number 2, I have dropped out number 2. I have tagged number 2 over here. From plastocurine, it will reach to the this one, cytochrome B6F, right? From here, it will reach to the plastocyanin, right? From plastocyanin, it will reach to the photosystem 1. From photosystem 1, this electron will be excited and received by P700. From P700, right, it will reach to the A0. From A0, so there is a electron carrier, that is A0. From here, it will reach to the ferredoxin, right? Ferredoxin, FD. Now, from ferredoxin, it will reach to FNR. This electron will be finally taken by NADP+. NADP+ will take electron, and this hydrogen, which came from the breakage of water molecule, this hydrogen will be taken over here. NADP will convert into NADP, what? NADPH, and formation of energy takes place. From 2 NADP to NADPH will be formed. I have already told you 1 NADPH is equal to 3 ATP, 2 NADPH is equal to 6 ATP. It means the energy equivalent to 6 ATP generated over here, right? Now, which are the electron carriers? Pheophytin, pheo means pheophytin, first one. PQ plastocyanin, cytochrome B6F complex, then plastocyanin, right? So this is all about the non-cyclic photophosphorylation. Is it clear or not? Tell me. Is that clear?
Yes, sir.
Right. Now come to this cyclic photophosphorylation. In cyclic photophosphorylation, electron will not come back. Look. Electron will start its journey from P700, that means PS1. It will get excited, right? Primary electron acceptor will accept. They will go to ferredoxin. From ferredoxin, when this process will take place, that is very important. That is a question for your NEET. When low light intensity is over there, low light intensity like 689, carbon dioxide fixation is inhibited, means stop, right? Or one more thing, when, let me write it down, when NADP plus is not present. When NADP plus is not present, then what will happen? There is no one to accept electron. When electron acceptor is not there, then what will electron will come back? Suppose that you goes to some places, someone is, you have been informed that someone is there to receive you. If that person is not there, what you will do? You will come back. The same thing happened with this electron. When electron acceptor is NADP plus is not there, it will come back to its home. So this from ferredoxin, it will go to plastocurine. When it will go to plastocurine, generation of energy takes place. ATP will form. It will go to cytochrome B6F, right? Cytochrome B6F complex. Again, ATP will be generated. Then plastocyanin, and the electron will come back again to P700. So this is called cyclic photophosphorylations. Cyclic because the electron came back to its original state. Then why phosphorylation? Because the formation of ATP is taking place. What is the speciality? When it will take place? When NADP is not present. Low light intensity. Carbon dioxide fixation inhibited. PS second only takes part in this reaction. In cyclic, both PS1, PS2 take part. Electron carriers are X or AO, FES complex, pyridoxine, plastocinin, B6F, and plastocin. These are the electron. Tell me, guys, go through with it and tell me, is it clear or not? Clear? Maimuna? Farad?
Yes, sir.
So that was the cyclic and non-cyclic phosphorylation. Chemoosmotic theory. Very important. Chemoosmotic. What is that theory? This theory tells how does the formation of ATP takes place due to transfer of electron. How does this process takes place? This chemoosmotic theory is given by Michel in 1961, and he explained how does the formation of ATP takes place in this light reaction, right? Or in this Hill reaction. So he said when photolysis of water takes place, hydrogen molecule is formed. It remains in the lumen. This is the thylakoid membrane. Look, I have written over it. Thylakoid membrane. And this part is actually the lumen of thylakoid, right? Water molecule breaks down, water is produced, and two hydrogen molecules are produced. Again, when electron goes to PS second, from PS second, this electron goes to plastocunine, right? From plastocunine, this electron goes to B6F. When electron goes to B6F, then due to transfer of electron, two hydrogen ions are pumped in again inside the lumen. Already there are more hydrogen ion. Again, the hydrogen ion are pumped in. Now the electron will reach to the plastocyanin. From plastocyanin, this electron will reach to the photo system first, then pyridoxine, then NADPH, finally take the electron. Now, due to this process, every time electrons are pumped in. So concentration of hydrogen ion, due to transfer of electron, hydrogen ions are pumped in. So every time when hydrogen ions are pumped in, the number of hydrogen ion or the concentration of hydrogen ion is reaching inside the lumen, right? Now what will happen? By this way, the concentration of hydrogen ion increasing inside the lumen, and outside outside, what is that? That is the chloroplasm, right? Chlorophyll protoplasm, right? So their hydrogen ion concentration is low, and inside hydrogen ion concentration is high. Means there is hydrogen ion gradient created between the membrane, right? Outer or inner side. Now hydrogen ion wants to, they will every atom want to diffuse from high concentration to low concentration, but hydrogen ion cannot cross this membrane. There is only one way to leak out or cross this membrane, that is this one, ATP synthase enzyme. I have told you the ATP synthase enzyme is responsible for formation of ATP. Now what will happen when these electron will hydrogen ion will leak out? Due to leakage of hydrogen ion, formation of ATP will take place. At the passage of every two hydrogen ion, one ATP is produced. Is that clear? Right? Clear? So one molecule of ATP will be produced when the two hydrogen ion passes. This theory explains how the formation of ATP takes place due to transfer of electrons. This is called chemoosmotic theory. This ATP which is produced over here is utilized in the Calvin cycle or dark reaction or C3 cycle. The NADP which is produced over here, it is also utilized in the Calvin cycle. So the product of the light cycle, product of the light reaction is NADPH and ATP. Where it is utilized, why it is made? Because this is had to be utilized in the Calvin cycle. Tell me guys, is it clear or not? Guys, come on, tell me.
Yes, sir.
Clear?
Clear, sir.
Very good. Excellent. Remember this theory have been given by Michel. This question have been asked. Is already asked. Hey, what is the Hill reaction? Even this question have been asked. Breakage of water molecule is called Hill reaction, right? Calvin cycle. Then now we come to the second half. What is the Calvin cycle or dark age, right? In Calvin cycle or dark reaction or C3 cycle. This dark reaction is also called black man's reaction. It do not require light. I have told you that's why the reaction goes on in day and night both. The reaction goes on day and night because it do not require the light. So dark reaction, it doesn't mean that it will go on the dark. Dark reaction goes at day and night. It is called dark reaction because it do not require light. This is very important. It was discovered by Calvin Melvin. That's why called Calvin cycle. It was discovered in chlorella and Sandesmus. What are these chlorella and Sandesmus? Who will tell me? What is the chlorella and Sandesmus? Any gentleman or any gentle lady? Anyone of you? What is the chlorella and Sandesmus? Guys, tell me. This is a very basic question. I need answer. Will tell me. Saifullah, Eva, Ellen. Tell me, guys. No one? Yeah, somebody has. Very good. Farad told me that it's an algae. Very good. So I'll add on that it's a green algae, right? Chlorella and Sandesmus, both are the algae, and they are basically green algae. And what have been used to trace it? Carbon 14 have been used in carbon dioxide to trace this cycle or to mate this cycle, to make this cycle, right? Now come to this cycle. What is this cycle? This is called C3 cycle, right? Means dark reaction or Calvin cycle is also known as C3 cycle because the first stable compound is 3 carbon, right? Phosphoglycerate. That's why it is called C3. This purple color molecule are the carbon molecule, the carbon atoms, and this purple color structure. This is the carbon atom, and this yellow color they are the phosphate. Now there is a first electron acceptor. There is a compound. No, name of the compound is ribulose biphosphate, RUBP. I want your 100% focus over here, right? One more important thing I will tell you. When this cycle revolve at one time or this cycle complete, it's one cycle, right? One round. At every round, it fixes one carbon, right? So glucose at every cycle, it fixes how many carbon atom? One carbon. Glucose is made up of 6 carbon atoms. So how many time cycle will run to make one glucose molecule?
6 times.
Very good. So to make one glucose molecule, the cycle have to go on 6 times. Right? It's very simple. Now you'll get it. The primary carbon dioxide acceptor or the first carbon dioxide acceptor is RUBP, ribulose biphosphate. Ribulose biphosphate is a 5 carbon compound. You can see 1, 2, 3, 4, 5, 5 carbon and 2 phosphate are there. This 5 carbon compound will combine with 1 carbon dioxide. So 5 plus 1, that is 6 carbon. So there are total 6 carbon. This reaction will means Rubisco plus carbon dioxide. This reaction will go on in the presence of Rubisco, an enzyme that is known as Rubisco. What is the Rubisco? Rubisco is the ribulose phosphate carboxylase oxidase. This enzyme help in the reaction with carbon dioxide, RUBP reaction with the carbon dioxide and with O2 as well. If the carbon dioxide concentration will be more, then this reaction will promote the fixation of carbon dioxide. If oxygen is more, then carbon dioxide fixation will not take, right? So because it carboxylize and oxidize both, right? So here the carboxylation is taking place because carbon dioxide concentration is more normally in atmosphere. So Rubisco, there is something specialty with the Rubisco. Can anybody tell me what is the Rubisco? Why the Rubisco is so important? Why it is very special? Any one of you? Why the Rubisco is so special? No one? Don't disappoint me. Write it down the question. Write it down the fact. Rubisco is the most abundant enzyme or protein. Make the star. Rubisco is the most abundant enzyme or protein on the earth. This is the most abundant. The only enzyme which is found in the maximum amount in this world. If I say which enzyme is present in the maximum amount in this world, then your answer will be Rubisco. And this is the protein compound. So Rubisco is the most abundant protein on the earth, right? So in presence of Rubisco, RUBP and CO2 will combine. They will make two molecules of 3 carbon compound. So 2 3 just 6, total 6, right? So what is that? This is the 3 carbon compound. Name is phosphoglycolate, PGA. Phosphoglycolate is a 3 carbon compound. This is the 3 carbon compound. That's why it is also known as what is also known as C3 cycle, because the first stable compound is 3 carbon. Then what will happen? This 2 molecule of phosphoglycolate, it will convert into G3P, glyceraldehyde 3 phosphate. It is also 3 carbon compound, G3P, right? When it will convert into G3P, 2 ATP will convert into 2 ATP, means energy will expend, means total 2 ATP will spend. Then 2 NADPH will convert into 2 ADP, right? Where both came from the light reaction. I have told you already that they are coming from the light reaction. They are coming from light reaction. So both are coming from light reaction and utilized here. Now 2 NADP which H will break down, phosphate will be released. Now what is this process called reduction? Now what will happen? This is a 2 molecule of 3 carbon compound. Now what will happen? How many carbon are total? Total 6 carbon are there. 2 3 just 6. Out of them 1 carbon is transferred for the formation of glucose. Now out of 6 only 1 carbon is transferred, right? Glucose, formation of glucose. Now how many carbon are left? Only the 5 carbon are left, right? Now the regeneration process, this is called regeneration process. Why? Because now the formation of ribulose phosphate takes place. Ribulose phosphate, right? Again the ribulose phosphate is formed. 1 ATP will convert into 1 ADP, means energy will be utilized, and 1 phosphate molecule will add on this, and formation of RUBP takes place. Again, the regeneration of RUBP takes place. That's why this step is called regeneration process, right? Third one, regeneration, right? So this dark reaction can be studied in the heading carboxylation, addition of carbon dioxide, reduction, addition of hydrogen, and or production of energy, then regeneration of this one. And every time this cycle will go on, and 1 carbon will be added to the glucose. Means to fix 1 carbon to make 1 carbohydrate, or to make 1 glucose, formation of sorry, the cycle will go on 6 times. Go through with it and tell me, is it clear or not? And let me know, is it clear or not?
Sir, can you say regeneration again?
Yeah, right. Look, this is the 2 3 just 6 carbon are there, right? So 6 carbon are total. Out of them 1 carbon is released. This carbon which came from the carbon dioxide, this carbon is removed. Where this carbon gone? This carbon which is removed from the G3P, it gone for the formation of glucose, right? So now how many out of 6, how many carbon are left over here? 1 phosphate and 5 carbon are left. So 5 carbon compound formation takes place, right? And the name of this compound is ribulose phosphate, right? Again, ATP will break down to ADP and phosphate. Energy formation taken place, and phosphate is released. The phosphate which is released, which will add on this ribulose phosphate, and formation of ribulose biphosphate takes place. This is called regeneration, because again the regeneration of rubisco have taken. Is it now clear?
Yes, sir.
So I have written over here again the equations. By the way, this is not required. The above cycle is sufficient. Carboxylation, that is RUBP plus CO2 in presence of ribisco. Formation of 2 PGA takes place. Reduction 2 PGA plus ATP. Formation of 1,3-biphosphoglyceric acid takes place plus ADP. 1,3-biphosphoglycerate, 1,3-biphosphoglyceric acid plus NADPH is equal to glycerol diide 3-phosphate G3P, right? Now regeneration, that is the formation of again this one, rubisco. Photorespiration. Can anyone tell me what is the photorespiration? You will not get this cycle, but you will get the question. You can get the question from this topic. What is the photorespiration? Any gentleman or gentle lady who is going to help me? Tell me. Photorespiration. No one? Okay, fine. So let me tell you. This is called C2 cycle, because first stable compound is 2-carbon compound. What happened? It is a process in which loss take place in photosynthesis. I have told you rubisco can react with the oxygen and carbon dioxide. If the concentration of oxygen increases in environment, or so how much it increases? Only 2 to 3% to 21%, right? So from 21%, 2 to 3% means it become 20, 23 or 24, right? So what will happen? Then this process will start. Now the carbon dioxide, look, in Calvin cycle, the carbon dioxide will react with this RUBP with O2, RUBP will react with O2, and it will make 2 compounds, right? So it what compound? It will make PGA and phosphoglycolate. By the way, formation of 2 PGA, 2 molecule of PGA should take place. But here the 2 molecule of PGA is not formed, because in presence of oxygen, RUBP have reacted with the presence of rubisco. So in presence of rubisco, oxygen, RUBP react with the O2 and makes 1 molecule of PGA and 1 molecule of phosphoglycolate, right? When it will take place, it is light dependent process. Release of carbon dioxide will take place from photosynthetic organ, right? This is a symptom that it is taking place. This question I've been asked. Release of CO2 will start from the photosynthetic or green part. When it will happen, it will due to increase of the intensity of light, concentration of oxygen increase, temperature increase, or other some factor, aging factor of the leaf. CO2 will react and it will make 2 molecules. 1 molecule of PGA and the another molecule is phosphoglycolate. This phosphoglycolate is 2-carbon compound. That's why this is also called 2CH. This phosphoglycolate will transport it into peroxisome. Here it will convert into glycolate, then convert into glyoxylate, then glycine. Now this glycine will be transferred to the mitochondria. In mitochondria, glycine will go and glycine will release 1 carbon compound and will become a serine. Serine will go back to the peroxisome. From peroxisome, it will convert into the glycerate. This glycerate will convert, it will go and again join this Calvin cycle. Here, the ATP loss is taking place, right? Here, the loss of energy also takes place, and the loss of photosynthesis also takes place. The process is called photorespiration. Photorespiration is a loss which takes place in C3, here the productivity of plant decreases, means plant will make less food due to photosynthesis. Tell me, is it clear or not? When and how it will take place, that is important. Photorespiration clear?
Yes.
Yes, sir.
Now let me tell you, this process will not go on the C4 plant. Means photorespiration, which decreases 20 to 30% productivity of C3 plant. This process cannot go on the C4 plant. That's why C4 plants are more advanced than C3 plant, right? What is the cringe anatomy? Very, very important. There are some plants, they are called C4 plants. These plants are immune to photorespiration. Means the process of photorespiration do not take place in these plants. They are called C4 plants. How they avoid the process of photorespiration? They have a different mechanism and they have a different arrangement of tissues. What is that arrangement? They have cringe anatomy. What is the cringe anatomy? In cringe anatomy, this is the structure of the leaf. In a structure of the leaf, at the at the mid vein, there is a vascular bundle, xylem and phloem. Around the xylem and phloem, there are very big size cells. These are called bundle sheath. They cover the xylem and phloem, bundle sheath. And above the bundle sheath, there are mesophyll cells. Mesophyll cells are those which are photosynthetic. So this structure has been enlarged over here. This is the mesophyll cell. It's a mesophyll cell. And this is the bundle sheath cell. Right? Mesophyll cell and bundle cell. Carbon dioxide will come and it will react with the carbon dioxide will come here. So fixation of Calvin cycle do not goes on here. Where? Here. It goes on in the bundle sheath. One more important thing. Where the Calvin cycle goes on inside the this bundle sheath cell are not permeable for oxygen, means oxygen cannot enter over here. Means where the Calvin cycle is going on, oxygen cannot. Means rubisco, where the rubisco is present, oxygen cannot go over there. So if oxygen cannot go, how the photorespiration will? So look. In C3 plant, please remember all those points. What I am delivering to you, what I am discussing with you. Look, in C4 plant, the primary carbon dioxide molecule receiver or acceptor was rubisco. Here, rubisco is not carbon dioxide acceptor. Here, there is a carbon dioxide acceptor that is called PP, PP carboxylase. In mesophyll cell, the enzyme PP carboxylase add the carbon dioxide to P. Now, the formation of oxaloacetate takes place. This is 4 carbon. First stable compound is 4 carbon compound. That's why this is called C4 cycle. Clear? Now this
Yes, sir.
glyoxylate 4 carbon compound will convert it to malate. Malate will be transported to the bundle sheath cell. In bundle sheath cell, malate will break down and the carbon dioxide will be released and it will be converted into pyruvate. Pyruvate will come back to mesophyll cell and again this pyruvate will become PP, right? And again the PP will receive the carbon dioxide. Here, this released carbon dioxide will be taken by the Calvin cycle. Rubisco will again take and it will start and make the sugar. Here, in the C4 plant, there is no chances of photorespiration, right? Why? Because oxygen cannot penetrate to those cells, that means the bundle sheath cell, where the Calvin cycle is going. So that's why by this way we can say the C4 plant are more advanced than C3, right? And this arrangement of the tissues is called cringe. Tell me, is that clear or not?
Clear, sir.
C4 cycle is also known as the Hatch-Slack cycle, HSK pathway. Named so Hatch, Slack, Korsack, they discovered the scientists. So that's why this is called HSK pathway. Uh, where does it take place? It takes place in tropical plants. That is also important. Or which of the following are C4 plant? You can get it. So tropical plant, they will be both monocot and dicot. Example, remember sugarcane, maize, and sorghum. They are the C4 plant. These all three examples have been asked in medical examination. So remember C4 plant. So they have the, you can get the question, which of the following have cranch anatomy? The answer is match. Which of the following will have which of the following are C4 plant? So sugarcane, maize, and sorghum, they all have cranch anatomy and they perform the C4, right? C4 cycle. So mesophyll shall have the chloroplast with developed grana. Bundle sheath will have the chloroplast without grana. So this two kind of the chloroplast are found. This is called dimorphic chloroplast. So C4 plant have dimorphic chloroplast. Why? There are two different kind of the chloroplast are found. How? In the mesophyll cell, the chloroplasts which are present in the mesophyll cell, they have developed grana. And the mesophyll cell, which are and the bundle sheath cell, which have the chloroplast, those chloroplasts without grana, they do not have grana. Right. So this is all about the C4 plant. Tell me, is it clear or not? Who will tell? All of you have to tell me. Is it clear or not? Tell me. Farhat, clear? C3, C4 plant? Saifullah?
Yes.
Hiba? I'll ask one by one. Hiba, tell me.
Yes, sir.
Ellen, Hanifa, Mamuna,
Yes.
Aksa. Say frankly, if you have any doubt, ask me. You are welcome with your doubt. No one have doubt. You all are very intelligent. But remember, right? Whatever I am saying, that example you need to remember. This example have been asked many times, multiple times. You will get this question previously. Okay, so that's great. Last but not the least, factor affecting the, it is also important. Factor affecting the photosynthesis. What happened with the light? One more thing. Law of limiting factor was given by a scientist named as the Blackman. That question have been asked. What is the law of limiting factor? The factor which is present in the least amount, will decide the rate of reaction. You are making tea. How many cup of the tea can be made? It will depend on the amount which is present in the least amount. Suppose that you have only 2 cup of the water. You cannot make the 4 cup. You cannot make the 2 cup of water, 2 cup of the milk. You cannot make the more than 6 or 7 cup tea. If you have only 2 teaspoon sugar, 2 spoon sugar, or 2 spoon the tea, whatever the thing which is available in the minimum amount, that will decide that how many cup tea you can make. So that was the law of limiting factor. So law of limiting factor says the component or the compound which is present in the least amount, it will decide, right? The compound or component which is present in the least amount, it will decide the rate of the reaction, right? Concentration of carbon dioxide, yes. If the concentration of carbon dioxide normally is present in the 0.3 to 0.4, right? If the concentration of carbon dioxide will increase by 0.05, it will increase the rate of reaction. But beyond that, it will not increase. Water, if water is less available, photosynthesis rate will go down. If water is available more, photosynthesis will be more. Temperature, it should not neither be more nor should be low, right? Go through with your NCRT and tell me if you have any doubt in this chapter. Guys, please go through with that. And when you will say that you don't have any doubt, then I will go to the next step. Clear or in doubt? Please have a glance with your NCERT. Go through with it. Okay, so if you don't have any doubt, then we'll move to the next chapter. That is a respiration in plant. Oh. Respiration in plant, right? The next chapter, plant zoology. It is also rich with information, so here also we have to focus more. Look, what is the respiratory substrate? So food burn in our body, right? Burning of food takes place in our body. Burning of food takes place in body of the plant. That is called respiration, right? So we do respire and plant also respire. So plant also releases carbon dioxide. We also releases carbon dioxide. But generally we say plant releases carbon dioxide and animal releases the oxygen. Plant releases the oxygen and animal releases carbon dioxide. Because the plant releases carbon dioxide in very small amount and they utilize itself and make the oxygen, right? That's why we say. Now look. Respiratory substances are carbohydrate, fat, protein, and organic acid. Burning of these takes place: carbohydrate, fat, protein, and organic acid. They burn. And to do burning of all these, so they can be means the carbohydrate can burn inside the body of the plant. They can generate even the same thing like the respiration part, same in the plant and the humans, right? There is a no difference, right? So respiratory substrate is that thing, that food which burned, and due to burning of this respiratory substrate, burning means respiration. Due to burning of this respiratory substrate, carbon dioxide is produced and energy is generated, right? So what can be respiratory substrate? It can be carbohydrate, fat, protein, and organic acid. Look, the carbohydrate, when we eat the carbohydrate will break down into glucose. Glucose will break down into pyruvate, and pyruvate will convert into acetyl coenzyme A. And this finally acetyl coenzyme A will burn and produce the energy. What about the fat? Fat will convert into fatty acid. Fat will convert into fatty acid. Fatty will acid will convert to glyceraldehyde. Acetyl coenzyme A, and again it will produce the energy. Burning of carbohydrate, wrote it down. Burning of carbohydrate and fat is called floating respiration. As a respiratory substrate inside our cell, if carbohydrate is burning or fat is burning, these burning of these two is called respiratory floating respiration. Now what will happen if the protein will burn? This is called cytoplasmic respiration. This question has been asked. Burning of protein is called cytoplasmic respiration. Why? Because if protein is burning means your protoplasm, your cytoplasm is burning. And if protein burning is started, then definitely the organism will die. That is for sure. The death of organism is for sure, right? So protein will break down into amino acid will break down into organic acid, and it will burn. Organic acid, they are the best respiratory substrate. They directly enter into, they directly get burned, right? So what can be the respiratory substrate and how does it take place? You know. Got it? What are the respiratory substrate? That I have told you that ATP will break down into ADP. Then yes, ATP will break down into ADP. Then energy will be released and phosphate will be released. Again, so this is called ADP. Again, if more energy is required, this ADP will break down into AFP. AFP. What will happen? Again, one phosphate can break down into one emergency situation, and the more energy will be released. AFP. And these are the phosphate bond, which are the breakable bond, and due to breakage of this bond, energy is released. So come to the next one. These were the respiratory.
Sir, 1 minute.
Yeah, please.
Okay, done, sir.
Types of respiration. And we know that the respiration are of two types: aerobic respiration and anaerobic respiration. Right? Very simple. When burning of food or substrate takes place in presence of oxygen, this is called aerobic respiration. Normally, this kind of the respiration takes place in plant or animal cell. So respiration or breakage or this burning process goes on inside the cell and within cell in mitochondria. So normally or in regular fashion or ideally, respiratory substrate is glucose. Rest are the respiratory substrate in some emergency situation or some special system. But the normally if we say a respiratory substrate, it means we are talking about the glucose, because the glucose burn regularly in normal fashion. So glucose will burn in presence of oxygen. Carbon dioxide will be released. Water molecule will be released. 686 kilocalorie energy will be. Now, anaerobic respiration. Anaerobic respiration takes place in bacteria, in fungus, and human muscles during the hard work. When we do hard work, running, exercise, hard working, then it also takes place in our body. Anaerobic respiration can be of. So anaerobic respiration takes place when oxygen is not available over there, right? Again, the burning of glucose will take place in bacteria and fungi. Burning of glucose will take place in presence of enzyme, formation of 2 carbon dioxide. Now, in place of the water, formation of ethyl alcohol will take place, and 59 kilocalorie. Now, you can see the difference here and there, right? Only 59 kilocalorie energy will be produced. But when this respiration takes place in human muscle, right? When this respiration takes place in human muscle, what happens? In anaerobic respiration in human muscle, the this carbohydrate, or we can say glucose, will burn. Formation of lactic acid takes place and 36 kilocalorie energy will be released. So you can just from here that in anaerobic respiration, the amount of energy is released, it is very less, very less. Now lactic acid formation will take place. This lactic acid will deposit in our muscles, in human muscles, and they will, the person will feel muscular cramp or muscular pain, right? So during the heavy exercise, during the running, the muscular pain takes place due to the deposition of this what lactic acid, right? Now what happened when we get exhausted? Every part of the body is like tired. We feel tired. It will like we are getting muscular cramp. We go to the bed or just sit, relax your body. Your pain will be relieved within 30 minutes. So what will happen? This lactic acid get deposit. This will be transported to the liver. There it will broken, right? So when lactic acid will be removed from that place, the pain or cramp will be relieved. So this is called types of. This is the called anaerobic respiration. So these were the type of the respiration. So tell me, guys, is it okay? Is it clear or not? And ask me if you have any question. Clear to all of you?
Yes, sir.
Next thing, respiratory quotient. From respiratory quotient, remember the value. You will get the question from the values. What is the respiratory quotient? RQ is equal to volume of CO2 evolved upon volume of oxygen absorbed. That is the formula. Respiratory quotient, if it is more, then this is the good one. Means this is the good substrate. If RQ is less, this is the bad substrate. Look. RQ for the glucose or carbohydrate is 1. RQ for organic acid is more than 1. That's why organic acid are the best respiratory substrate. Let me write down. Best respiratory substrate. Fat, it's 0.7 that have been asked. Protein 0.9 that have been asked. All both questions have been asked, right? What is the compensation point? One more point, important, very important for you. What is the compensation in a photosynthetic cell? If release of oxygen is equivalent to release of the carbon dioxide, right? It means neither carbon dioxide is released nor oxygen is taken. Why? Because during the photosynthesis, the oxygen is released. That oxygen is utilized by the respiration, and the carbon dioxide which is released during the respiration is utilized by the photosynthesis. Means when there is no evolution of oxygen and no absorption of carbon dioxide, this point is called compensation point. In a photosynthetic cell, when there is no exchange of O2 and CO2, because the amount of the oxygen is equal to the amount of carbon dioxide is produced, that is called compensation point. Tell me guys, is it clear?
Sir, can you scroll up, please?
Yeah. Clear?
Yes, sir.
Okay. This is very, very important PPT. I have made it for making you understand. Try to understand. Like, how does this? There are a step of it. What happened? We eat bread, we eat rice, we eat number of the grains. They all are carbohydrates. Means all are basically starch. Because plants store their food in the form of starch. This starch will go inside our stomach. Digestion will take place. Digestion of carbohydrate will take place. These carbohydrate will be broken down into what? They will be broken down into glucose. Now this glucose will be absorbed by intestine or villi. Then villi to blood vessels, and from blood vessels it will dissolve into blood. So finally the glucose will dissolve into the blood. Now this glucose will be transported to all the cells, because every cell of the body is require the glucose, right? So now the glucose is transported to the cell. Now glucose went inside the cell. Inside the cell, glucose will be broken down. It will be converted to pyruvate. Why pyruvate? Because glucose cannot enter inside the plasma membrane. Sorry, inside the mitochondria. This is mitochondria. Finally, the burning will take place in mitochondria, but glucose, unfortunately, glucose cannot enter inside the mitochondria. So what will happen? Glucose will be converted to pyruvate. Breakage of glucose into pyruvate takes place. Glucose is 6-carbon compound, pyruvate is 3-carbon compound. Breakage of glucose into pyruvate is called glycolysis. This process is called glycolysis. During this process, ATP is produced, and the process takes place in protoplasm, cytoplasm, right? So glycolysis, during the glycolysis, formation of 8 ATP will take place. 8 ATP, net gain of the 8 ATP will take place, right? Now it is converted into pyruvate. Pyruvate is 3-carbon compound. Now this pyruvate can easily enter into the mitochondria. Now pyruvate is 3-carbon compound enter into mitochondria. But inside the mitochondria, this pyruvate cannot go inside the Krebs cycle because the burning of substrate will finally take place in Krebs cycle. This pyruvate will be converted into 2-carbon compound acetyl coenzyme A. Now this acetyl coenzyme A will go into Krebs cycle. Krebs cycle will go on. Complete the burning of substrate will take place. Carbon dioxide and ATP will be released, and the energy will be released in the form of ATP, NADPH, and FADPH. They all are the energy molecule. They will be generated, and carbon dioxide is produced. This is the complete process of the respiration. Did you got it or not? Is it clear? Farhat,
Yes, sir.
Abdullah,
Yes, sir.
Iba,
Yes, sir.
very good. Ellen, Hanifa,
So,
very good. Right. So at least take the screenshot of this one. I ask you to please do remember and this make it on your notebook if you have time. Right. If you don't have, then it's okay. You can read from the screenshot only. Right. And do all the questions in 2018 onward. The questions from the that transport part. In next class, I will teach you the glycolysis, Krebs cycle, and then we'll finish this chapter. Right. And ask me if you have any doubt. Tell me if you have any doubt, any kind of doubt. Please tell me if you have any doubt.
Sir, how much ATP is going to be produced in total?
And total 38. Very good question. And if there is option 36, don't take the 36. Sometime you will get 36, 38 both. 38 is the correct answer. And in internet, there are number of the time there is a mistake. Some questions, there the option is 38, 36 is also option. And that is correct answer. So 36 is not correct. 38 is officially I declare 38 is correct. Right? And in NCERT 38 is given. Okay. 13 Krebs cycle, 8 in glycolysis, total 38. Clear?
Yes, sir.
So let's meet in the next class, right? Bye bye. Take care. Have a good day.